is we're going to talk about this shot. So let's just go over here and see if he's over there because I think I can hear him. There he is with the
Hey,
background and everything. How the hell are you?
good, good. Sorry for that fumble on my end. I was on Google Meets instead.
Dude, it's
Looking
it's
for you.
it's really okay. But really, thanks for taking the time to talk to us. So you have been pushing this message of the idea that the RNA isn't all what it's cracked up to be for quite some time now. You just got kicked off Twitter today, not for that though. Is that correct? Something else got you bumped?
Well, I don't know 'cause they forwarded a tweet from Monday. Although they gave me a strike within 10 minutes of posting something like last Friday. They were hot to trot on that one, and then this one I think they dug up because an article just published that I will forward to you maybe in the chat about Alex Berenson and Andrew Bostom and a few others of us who have been getting deplatformed.
Oh, are
And
you
that
also
one,
in there? Are you also in there?
yeah, I'm in there, like heavily
Oh,
in there, attacking the NIH about their ridiculous tweet this week.
I'm gonna put that up right now. You sent it to me just before.
Yeah.
Okay, wow.
So the NIH comes out with one of these, unicorn posts saying, "Hey, that this
Oh,
doesn't
there it is.
integrate."
Look at you. Oh my
Yeah.
gosh.
So they waxed me right after that went live.
That's hysterical. That's like blatantly like that's just
Yeah,
weak. That's like
it is.
teenage level.
And the fact that they had to dig to Monday to find something means they dug. Because I've
That
clearly
is
posted more stuff since then.
hysterical.
Yeah. But yeah, that one is, NIH trying to claim that the mRNA is no different than a natural mRNA, nothing to fear here. It doesn't integrate to your genome. And so I didn't even hit them on the genome integration because I understand that's a bit more controversial. There's two papers that people refer to, which they didn't even acknowledge. They just said, hey, it doesn't integrate.
Yeah.
And frankly, the NIH should be like just play neutral right now because it's an open debate. You've got the Whitehead paper that shows some integration. People are questioning the methods and whatnot, but it went through I think 2 rounds of torture. The preprint got tortured, and then it went through PNAS, and they addressed a lot of the concerns over the cDNA step, perhaps causing some artifacts. And then there's another one that's more recently just showing that it does reverse transcribe, presumably due to line elements, but they didn't show integration in that one. But
Right.
the two papers combined kind of hints that yes, it might happen. But we don't know the frequency. We don't know if it's getting to germ lines. It could just be a couple cells that get this, and maybe it's not a big deal. We don't know. But to come out and flatly say it doesn't happen and not reference those two papers is weak. But I didn't really hit him hard on that. I went after the fact that you can't claim it is an mRNA that's completely natural, particularly when you're in a patent dispute with Moderna over your invention claiming it's not natural.
Yeah, right. Yeah.
The whole pseudouridine thing isn't, yes, you can find pseudouridine at low frequency in mRNAs. It's 0.2 to 0.6% in the literature. It's not 100% of the uridines being replaced. The two prolines are different. They changed those. So there's just so much hypocrisy going on. Them trying to claim to the USPTO that, hey, give us a patent on this. It's not man-made. It's been modified by man, which is the only way that you get something like that to be patent eligible. And then to turn around to the public and say, oh, it's the same. That's either your patent should be null and void, or you should own up to the fact that there's pseudouridine in these things. And for someone like the NIH, who funds so much genomics, to get that wrong it begs the question of why are they doing this? Like, they know this isn't true. They're just pimping a vaccine, which they're in a patent dispute over who gets the benefits of. The royalty stream coming in from this could be $5 to $10 billion.
Yeah, it's
So
it's it's definitely one of the main things that we need to constantly pay attention to is following the money.
yeah,
Um,
yeah. So I just that's blatant. And so I point out to Twitter that if you're going to suppress people correcting the NIH from making these statements, then and they're doing this because they have patent royalties. This is like racketeering.
yep, yep.
Like they're getting themselves into a total mess. And Elon shouldn't go near this thing because if this all does blow up, the class action lawsuits are going to drag them into it.
Wow, I just didn't. I guess I just didn't realize that you had obviously that you had been quoted, and that would be directly retaliated against by Twitter.
Yeah, I poked them pretty hard in the eye on that one, and I think they just when they saw that they must have gone through my profile and just found something to. And the one that they went after, I mean, God, I don't even think that one's factually inaccurate. I said the vaccines have not. No one's proven a reduction in hospitalization and death in an RCT. I don't think that's factually incorrect. I mean, am I off on that? Have you
No,
found
it's definitely not factually incorrect. That was the reason why I was having trouble figuring out what exactly happened today. So when you said that you were kicked off, it didn't make sense.
I mean, maybe I should have been more specific, maybe saying that Pfizer and Moderna never did it, because I don't know about every vaccine trial on earth. I just know that the dominant ones that are getting shoved down our throat never showed reduction in hospitalization or death. And not in the RCT. I mean, there's a bunch of other papers that Pfizer's like funded, and the PIs have money from Pfizer, and all those things are like have so many confounders in them that they just don't hold up to the data that we're actually seeing now, which is that the vaccinated seem to be driving this. The most recent Omicron, like whatever it is, BA4 and 5, are the numbers imply that you're at a higher risk if you get boosted for this thing.
Absolutely. I mean, I don't have any doubt about that anymore. That was something that we had imagined on the back of a napkin a year and a half ago would happen when they immunized only to the spike protein, and now it's happening. And the protein that's changing is primarily the spike, and so everything just jives. I mean, it's definitely feels as though it's now pretty certain that the transfection is what's driving this changing spike protein, and as long as your immune system is not focused on the spike protein, you're probably going to be all right in the short term.
That's another thing they dinged me on. One of my other tweets was speaking to the fact that it was transfected, and they took offense to that as well, saying, this is basically is not like any other vaccine. It goes into your cells and gets expressed. This transfection is not what we see with other vaccines, and you can't claim the spike is innocuous from all the literature we have on SAB and people showing the spike has some toxicity. So I'm not even cited Chang at all on that, and they still whacked me with peer-reviewed citations on what I'm saying.
Well, I think the way that I would like to approach trying to summarize your current, the stuff that I think you're doing that's most important with the RNA, and trying to really explain to people the differences between what they purport to have done and what they have done, is first of all to remind my viewers, and maybe you haven't seen it because it's just one of these dumb things on the internet, but there was a video a month ago or so by the lady who works for Moderna, and I don't remember her name, and I wouldn't want to say it anyway if I did, where she explains that Moderna has been working on a number of ways to use mRNA in medicine, and most of those ways involve expressing endogenous proteins for which there is a genetic error. And so replacing an enzyme in the liver is one of the lowest hanging fruits they've been trying to hit. But in this explanation, she very cursorily goes through discussion of that to the mRNA vaccines for the mRNA transfection for coronavirus, without really pointing out that there is a biological Grand Canyon between expressing an endogenous protein and expressing a foreign protein in the sense of how the body will react. But then even more so, she, in my mind, as a salesperson, she glosses over she doesn't mention it at all, which is where your biological, expertise comes in the most. In the ideal situation, they are using an RNA with 100% fidelity. So the RNA goes into the cells that they want it to, and it expresses the protein in the exact form that they want it to, with no mistakes made in any of the products that are produced. And it lasts for the exact amount of time that they intend it to last for, and your immune system learns to respond to that protein in the way that they want, and then you're immune to coronavirus. So the last time we talked, we were really focused on this G quadruplex formation.
Oh yeah,
but
what might help? I did recently this paper at Panda, so I have some slides that might help fill in the gaps.
I looked at them briefly, so you can definitely try them again. I don't know why
Um,
you're locking up here, but hopefully this will go away.
that could be me. What do I have? Yeah, internet unstable. We are in like a brownout right now in where I live. They've asked us to everyone
Okay.
turn off the power from 5 to 7. Drives me nuts. How's that? Is it coming back? Oh, you know what I should try and do? Let me just
It's okay.
nuke everything else on my computer.
I have some. I took some of those slides and pieces of them as well, but I don't mind if you want to present. That's I will just.
We can
That's
pay if you want to page through and ask questions. We can do that too. I don't want to death by PowerPoint your audience here.
no.
Um,
I don't really mind. So the main, I guess the main thing that I thought that I found from your panda discussion that I hadn't heard in our previous discussion. So of course we've talked about the fact that the RNA will fold differently. This is the G quadruplex idea.
yes. Yeah.
And that folding differently will cause it to be can cause it to be regulated differently. It can cause it to be spliced differently. It can cause it to be translated differently.
Yeah, and it may even be causing their transcriptional stalls as well. That's something that is it's in the recent EMA leaks, right? So those earlier EMA docs pointed out that they had RNA integrity issues. I've never seen one of their adjuvant traces for this, but they had a set threshold of you have to have over 70, higher than the 70% RIN number. It's an RNA integrity number you get off of these like adjuvant stations that measure these things. And then the production batches fell down to 55%.
Yes, and they accepted them.
Through.
Yes, they pushed them through.
Anyway. Right. So why does RNA fragment? I mean, yeah, it is a molecule that you can shear if you're pipetting it too fast, or if you have RNases or other impurities around. Even metals and heat will fragment RNA. But the other thing that will create truncated transcripts like that are quadruplex G structures. The T7 RNA polymerase can't necessarily push through those things, and so it makes stall points.
And so you're talking, just to be clear for the people who are listening, you're talking about during the manufacturing process, right. They're using enzymes to make copies of the RNA that they're eventually going to inject in you.
They are. They put the construct into a DNA plasmid so it's stable and they can grow shitloads of it. Then once they purify that, and they got to get all the LPS out of there when they purify it to make sure there isn't any lipo, any of that stuff around that can cause an immune reaction. But they then run T7 polymerase over it, where they rip uridine out and put in N1-methyl pseudouridine. So they have this alternate base, and they beg the enzyme to put that in, and that's where the enzyme makes a lot of errors. But you have to realize the template that they're going off of right now is probably all knotted up because it's now loaded with quadruplex Gs on the DNA level that it's reading off of. So it's going to stall on that and give you truncated mRNAs.
So this is separate. This is a G quadruplex problem in the DNA, which is producing the RNA. This is separate from
I wouldn't be surprised if that's why they have truncated products. So those are in the enzyme stalling on that. Probably what happened is when you scale these things up, you have to scale the nucleotide synthesis up. So the N1-methyl pseudouridine probably had to get scaled to gargantuan synthesis scales, and the purity of that is always a problem. And if its purity isn't right, you can be putting in some of the synthesis analogs instead are in there, and the enzyme can stall on those or run into the quadruplex Gs, and you end up getting truncated transcripts, and they can be tricky to purify out unless you have like a biotin tag or something on there, like a poly A tail that you put on. It's hard to fish them out. Now, when they're going from 5' to 3' synthesizing these, they don't get to the 3' end unless the transcript goes all the way to the end. So they probably have some type of poly A hook that tries to pull these out, but those don't always, they're clearly they're not getting full length product, which is why it failed that metric. But the EMA got apparently coerced to push it through anyway. So now we've got shots that don't have full length mRNA. So the question is, one, those probably aren't going to fold. Like when they make an actual protein, it's not going to be full length. So all bets are off on that folding into a spike protein. Now, is it innocuous? I mean, it could be that, okay, you have something that can't form adequate immune response, and you're getting all the downside of the neutropenia and lymphocytopenia that you get from injecting these LNPs. So what was evident in the Pfizer trials and the AstraZeneca trials is that like 46% of the patients, I think in the AZ trial, had neutropenia, and almost a similar number in the Pfizer trial had lymphocytopenia. That's like half the patients having white blood cell depletion after like 7 days after injection. All right, so something about the LNPs is wiping out the white blood cells. So you're actually more susceptible to other infections in that first 7 days, which is why they played this whole game of you're not vaccinated until 14 days out, because they know.
Why do you think it's the LNP and not just these proteins that are
Well,
being
it's a good.
produced?
I don't have firm proof of that. I've just seen a few papers that suggest the LNPs alone can be immunogenic. In fact, this came up with that wonderful discussion we had with Debunk the Funk, where he was trying to make the claim that the HPLC is immunogenic and the N1 pseudouridine is immunogenic. He had it completely backwards. Those are the things they do to make it not immunogenic.
Right.
And he forwarded a paper which we clearly pointed out to him that no, that paper says the opposite. The actual LNPs are causing the response, not the mRNA.
Right.
And it's not double-stranded. They HPLC this to make it single-stranded. That was funny because even Jay Bhattacharya he texted me in the side, and I'm like, why is this guy saying the exact opposite of the papers he's publishing? Like, I'm no molecular biologist, but I read that. I understand it's the exact opposite of what he's saying.
Yeah.
So there's one paper linked in there, if you can get to the tweet anymore, that goes over some studies they did where they injected just the LNPs and just the RNA and then combinations of both, and they can see which ones are driving more of an innate immune response. But I'm assuming that's maybe what's driving the lymphocytopenia and neutropenia. So if you're going to take this hit from the vaccination where it weakens your immune system for a brief period of time in order for you to build the antigen, it'd be a real bummer if the antigen didn't build correctly, which could be what's happening batch to batch or lot to lot, that they just don't have this RNA integrity under control. And so some people are getting blanks and making misfolded spike proteins, which we don't really know what that does. And then the other thing I've often considered, because I haven't seen any of these adjuvant traces that are describing this RNA integrity number, but having used those adjuvants, we've got one in our lab, the RNA integrity number can also be off if the transcripts are larger than expected. Like what RNA integrity is looking for is a really clean peak with no scatter on either side of it. And I've seen some of the other posts, some of the people in your community pointed out that if this thing reads through the stop codon, there's a frame shifted GP130 there that they could read. And if it just keeps going straight onto the same transcript, it's going to read a poly A into like a polylysine track or something. So that would be ugly as well. So we really need to see these traces to know, is it just fragmentation or are there larger mRNAs that are coming out of this that end up in there as well? Certainly when these things are expressed, we're seeing very smeary gels in a lot of the studies that look at this, suggesting that, and then when I say expressed into proteins, these are Western blots that are showing protein bands that aren't a single band. And they tend to blame that on, it's the glycosylation peak. But they're only like maybe a half a day away from testing that you can use an enzyme that rips that off and to test it. And it's unlikely that you would get that many Daltons of a shift from glycosylation, because it's rare that you get 100% glycosylation. You might get 5% glycosylation. And so it wouldn't add up to like a 30 kilodalton shift. So they're doing some hand waving on the glycosylation excuse. They should really be demonstrating, like you can use an enzyme PNGase to like test like with and without that. What's the gel shift look like? And the fact that like 1 day experiment isn't done is really suspect. That's not hard for them to ascertain.
There are quite a number of 1 day experiments that haven't been done, right? I
mean, right.
There are quite a lot of 1 day experiments.
Yeah, like sequencing the damn thing.
Yeah,
that would be a miracle, right? Well, yeah, I don't know where. Well, why don't I at least hit on the codon optimization area, because I think some people have asked, like, what is that all about? What's going on there? I can hit. Let me see if there's how would I share this on your thing? Let me see if share screen right down there. Bingo. Oh, I think you got to enable share screen.
And this one, and this one, and this one. There you go. Should be good.
All right, let me hit this thing up.
So codon optimization is for you, is it also relevant for your work? Do you codon optimize things for expression in cannabis plants? And is there a different kind of set of codons for them? Like,
Yeah, we absolutely do, but we do that when we're going to slaughter the host.
oh,
It's
I see. Okay.
like if you don't care about the E. coli and you're going to lice them open to harvest all the protein you can out of them, yeah, we'll codon optimize. We did
Yes.
that. We found a thaumatin-like protein that seems to attack yeast and molds, which we thought was driving the powdery mildew resistance in cannabis. So we cloned it into E. coli, codon optimized the thing, and got gobs of protein. But we're not really paying attention to whether the E. coli is going to survive that. It's just we're trying to crank it to the highest expression level possible because we want a lot of the damn protein.
Right.
There's a his tag on the protein. We can purify the protein out afterwards. There's all these things, these other tricks you do. But that's making the assumption that maximum expression is desired in a health circumstance like this, which I think they've never demonstrated that maximum expression of protein is the best way to generate a response, considering spike has some concerns of its own toxicity, right? And the other thing to consider is like this is a virus that has evolved in its host. So it has selected these codons probably for a reason, because they're not the optimal codons you'd use in a mammalian cell, right? And the reason they codon optimized is said, hey, it looks like it's broken, we should fix it. But maybe the virus actually evolved this codon usage so it would get the right balance of expressing itself and being able to transmit with a host that's not dead. This idea that, we should just codon optimize it and go for as much as we possibly can is kind of rooted in, I don't know who thought that up. It's like, yeah, that's true if you want to make gobs of a protein in an industrial fermenter that you're going to kill all the cells and you're done, but these are humans. And if the virus has evolved this certain codon structure, it's probably done it so it precisely folds the enzyme or the spike protein in a way that is most judicious for survival of the host and passage of.
I think it's a really wonderful point.
Yeah, there's a little bit of lack of humility here, I think, and hubris going on, that they're thinking just turn this to 11 and all is going to be fine. Now, so anyway, that's kind of what that slide is meant to do. So for people who don't understand these codon optimizations, what you're seeing in these codon tables is all of the amino acids that certain codons code for. And you'll see there are some amino acids like arginines, in the discussion quite a bit because there's some bizarre codon structures in arginine in the virus, but there's 6 different codons for arginine. All right. Now, the virus never uses CGG. Humans do, and which is what stood out to Kristen Anderson when he emailed Fauci saying this doesn't look right. The furin cleavage site is already using human codons for arginine, and nowhere else in the virus is it doing that. What the hell? Who put it there? So that's right next to the SEB domain, which I think is important to touch on as well. I think your audience probably knows what that is and knows the furin cleavage site is kind of one of these Dazic smoking guns. But anyway, so cannabis and E. coli are going to use very different codons. When a virus that is infecting humans decides to settle on a certain set of codons, you probably don't want to mess with that. Because it's probably, particularly since SARS-CoV-2 decided on a set that's not what we have anticipated as being humans. We would have selected different codons for this, but the virus decided to use really inefficient ones, maybe because it needs them to be that way. And there's in fact some papers that demonstrate when you move these things around, you get different ribosomal pausing and you get different folding of the protein. In fact, the paper that talks on this, I think I have in here, actually nails this right in, it happens actually in the furin cleavage site, which is kind of fascinating. All right, so there's a couple problems when you codon optimize. The GC content changes. That means the RNA is going to change. The RNA sequence is not the same. NIH, you should know this. When the GC content changes, this has lots of implications. The RNase is going to change. The microRNAs that hit this thing are going to change. The toll receptors that look at these things are going to have an impact. The ApoBec biochemistry is going to change. So you don't just change this and say this has no impact, right? That's very, I think, high school biology. Like here's your codon table, you can predict everything you get out. No, there's a lot more complicated circuitry going on with these RNAs, And when you start messing around with the GC content, the microRNAs suddenly don't behave the way you thought they would.
Now there was a paper in I think 2011 in Science where they started to admit that the silent truth of silent mutations was that these mutations, even though they're synonymous mutations, can
They're
be
not so silent.
they're not so silent, and they can underlie disease states because the protein folds differently. And
Yes.
that was something that up until, not too long ago, people really, really ignored. In 10 minutes, we'll either have to switch or you'll have to be done, but that's okay. We can
Okay.
just
All
switch.
right. I'll go fast.
It's all good.
Yeah. So yeah, this can alter the translation rate and protein folding, as we just said, and it creates these quadruplex Gs. That can be a problem. But you're right. The real thing we should be cautious about is that we don't really we can predict when you change a coding base that's going to change the amino acid. We got great tools that can predict that, but the silent ones are really still idiosyncratic, if you will. We don't really understand how to predict what those things are going to do. We usually just assume they don't do anything, but when you change them all in the entire transcript, you're getting into some really unknown territory. So this is the GC content changing. You can see this in the paper, and if you put them through mRNA fold, you can see that the spike protein in the the spike sequence, the mRNA sequence folds differently. All right, so if you put the DNA in there as well, RNA fold and other folding things will show you the different knots that you get with double-stranded DNA. I wouldn't be surprised if some of those knots actually line up with where they have some stall points in their T7 polymerase trying to express RNA off of it. So I point people to Postikova's work. They actually looked at the furin cleavage site and showed that when you start mucking around with those codons, even silent changes like this, you end up changing the ribosomal pausing rate across that, and the protein folds differently. So we already know in one codon, a very key one, that when you change this, things change.
You know what's so crazy about that guy, the debunk the funk guy? His only publication is about mutations in the small subunit of ribosomes and how it affects translation. The only thing that guy should really understand is how screwed up RNA of this magnitude could be, and how it would be intimately affected in terms. I mean, it's his expertise. It's the only thing he should know after his PhD, and he's like completely ignorant of it, as if there's nothing to do here.
Yeah. So if you look at G4s in the virus, they are highly conserved. There's one paper that looked at 16,000 different SARS genomes, and the G4 regions don't change. So they're clearly playing some type of regulatory or structural role if they're conserved. And they just added like 19 of these things into the RNA willy-nilly, not thinking twice about it. I think we have that here. It's in the paper. I'll skip this and go fast because you can get this off of a preprint. But we just highlight where all the G4s are using this common GQRS as a tool. You can do this yourself. Take the RNA sequences, plug them in there. You'll get the same results. They highlight where all these G4s end up being. So these are areas of knots that are going to exist in the RNA, and they'll also exist in the DNA transcript that they're trying to code off of. All right, so when you change N1-methyl pseudouridine, this is a paper that looks at just changing a few of them. I think we're looking at 1, 2, 3, 4 here. Just 4 of them in the sequence, and the TM shift jumps like 9 degrees centigrade. That's enormous. Now
that's the melting point.
That's the melting temperature. Yeah. Yes. So this radically changes the way base pairing occurs, which means when your tRNAs go to try and read this stuff, they're going to be really confused because the melting temperature of these things is all different. Now, there's something to know. Pseudouridine and N1-methyl pseudouridine are different, and that 1-methyl group does change base pairing a little bit. However, the TM goes up for both of them. When you have that methyl group removed, there's one more hydrogen available for some funky base pairing. So pseudouridine is worse than N1-methyl pseudouridine, and you'll see that in some of the error profiles, but they both create errors, more so than native. I probably don't have much time to go into this, but this is for people's reference. There are other biological switches that are very sensitive to these things. MicroRNAs and other are very sensitive to changing these methyl groups around. But here's one of the really good papers showing stop codon ablation when these pseudouridines are in place. All right, so the tRNA has to anchor on just 3 nucleotides. All right, so when you alter them, that's a lot of change for a very small target. And so this is where they're seeing a lot more error rate in the translation of these things. And we've pointed out that they used UGA stop codons, which is probably the worst they could use. They're most likely to frame shift and be read through. They doubled them up, which shows that they kind of knew this was going to happen. And the regions after this are here. If it frame shifts, it makes this frame 2. And if it doesn't frame shift, it will continue to read through the stop codon. You'll get this other protein. Not much on the green one, but the red one, we can find GP130 homologies to. So if it does frame shift, it's going to make a chimeric virus human peptide, which could create all types of autoimmunity hell.
Why would that even be there?
I don't know. I think it's just a spacer until they get to their poly A. I don't know why that's there. There are some very mysterious things, but this is the Pfizer one. And interesting enough, in the Pfizer one, the longest ORF is actually on the reverse strand going the other direction. It doesn't have any promoter or 3' UTR for it, but it's bizarre that they actually codon optimized this and got a completely in-frame ORF longer than their target ORF on the reverse strand. That's really hard to do. I think James Lyons Wheeler has like a little presentation on that, but
I need to look at that.
Yeah, that to me was like, whoa, how did that happen? That's usually you get all types of stop codons riddled when you do this. Some other citations for people who want to just quality check what I'm doing here. This is where I've been pulling some of these frame shift issues from pseudouridine, so people have access to some of the references. But this is, I think, is the most frightening number here, is that when they just change these around in a single codon, not changing all of them in the mRNA, just a single codon, you start to see error rates build up in the translation products. And they're putting this at a pretty high number, 1.5% integrated over all these codons. So that's not a small number when you're putting in trillions of these things. All right, you're talking about billions of problems that could be making misincorporations or mistranslated products. That's one form of error. The other form is at the translation step before you do this. When they're making the mRNA, before they inject it into you, they have to make it off of that DNA, and they're going to use T7 polymerase to do it. And it's going to make a shitload of errors in the process because it doesn't like to use this dirty base, this N1-methyl pseudouridine. This paper would use some really nice packed biosequencing to measure it. And yes, you can see pseudouridine has a higher error rate, but methyl-1 pseudouridine is right there as well. So it's almost doubling the error rate. And these error rates, if you calculate them, are 200 to 300 per million, which means about 1 in every single VAX molecule is wrong. There's an error in every one of them, probably. This is a compounding error. You have this error going in, saying there's just simply misincorporations going on at the transcriptional level, and then you feed it to your tRNAs and your ribosomes, and then they start making 1.5% errors. So you begin to understand why they've never given you DNA sequence, and they've never given us protein sequence of what this thing makes in human cells. That's been hidden. And I think it's because it's a big goddamn mess.
Where do you think that at the original manufacturing level from DNA to RNA, it's a significant problem already? And then
yes,
yeah, okay,
yeah. I mean, you don't want to have an error in every molecule, and you inject 40 trillion of these things, and you have a super antigen in there. You're running a mutagenesis reaction on a super antigen across 1 billion people. That's fucking insane.
Yeah, I don't even know I don't even know really how to process that, because we're not even talking about. Yeah, we're not even.
I mean, the way some of the papers like nerf this stuff is they're like, oh, that might be great for antigen diversity. It's like, come on, this is reckless. You can't do that.
It's extremely reckless, especially because they want you on one hand to believe it's a bioactive protein, and on the other hand, it's fine. And we know it's a bioactive protein. We know that it has all these added attributes. We don't really know how to adequately describe it, but.
Now, one critique that we've been levied against us quite a bit is this is all theoretical. You guys should pick with pipette, all that stuff. Like, right, Pfizer's going to give us vials to test for this, right? We have to go dumpster diving. But one thing that did come out this week, I sent you a link to out of the Patterson's lab. His Twitter handle is Dr. Yo-Yo, I think. I think his last name is, I'm going to murder his last name, but they do great work, excellent work, and they're often not getting credit for it. But they demonstrated with mass spec that they're finding mutated S1 proteins in the vaccinated, but not in the long COVID patients with C19. Right, so right there is evidence that the vaccine is producing error-prone peptides. And now I've been trying to map those mistakes that they found to certain codons in quadruplex Gs. I haven't quite got it all worked out yet, and I'm not certain it necessarily correlates with codons that would have pseudouridine in it, or that it would be near G quadruplexes. But nevertheless, there's errors documented in mass spec and vaccinated patients showing the spike protein's mutated. It's a preprint. I'm sure it's going to get murdered, but it's out there. I think we're running out of time here.
No,
but,
I just slapped it up. It's okay. I mean, I think this was a really good, sharp revisit of where we've been. What the hell just happened there? Where did you go?